So, let's see if this works. Alright, so this is the opening picture to Chapter 9. So for our genetics topic, we started out in Chapter 7 with the basics, right? And then we're going to, the rest of it, we jump to Chapter 9, where we look at more genetic analysis. So in this picture, you have two bacteria. Can anyone tell me what this is representing in this cell? The nucleoid region. What's in the nucleoid region? What would we say this is? DNA, right? And when it's in this form, we call it the chromosome, right? And we know for bacteria, how many chromosomes do they have, typically? They're not an abnormal fact. One, right? So this is one long piece of DNA hooked back on itself in a circular way. And then it kind of folds up on itself, right, and compacts. Right, so it looks like a squiggly mess. This other guy over here, not all bacteria or prokaryotes are going to have it. What is this called? A plasmid. And it is DNA as well, and it's circular as well, typically, right? Unless it's abnormal. So it's double-stranded, hence the two colors if you can see it. Okay? And not all bacteria that are going to have it. But it is DNA, so it is information. So in this process, notice there's this, I don't know if I can reach that high. Oh. See that little thing coming out? All right, these two bacteria are hooking up with each other. And that plasmid is actually going from one cell that had it to one cell that didn't have it, right? And now look, he has it, and it gives him the ability to make this little thing that's sticking out. This is one of those touchscreen things. Okay, I will stop touching it. Okay. It can make this little thing that's sticking out. Okay. What is that little thing that's sticking out? Anyone know? That little protein appendage. It's actually a hollow protein tube that's connecting these two cells. Sex papillae, or papillae, right? So this allows bacteria to do a particular process that sometimes is referred to as bacterial sex, because that's what sex is exchange of DNA. But in this case, it's given a different name, starts with a C. Conjugation, right? And other organisms do this process as well, right? That we know as conjugation. But bacteria can do this, and this is one of the ways that information can go from one bacterial cell to another. And the unfortunate thing for us is a lot of times, some of those plasmids are called R plasmids. Anyone know why they're called R plasmids? It stands for resistance. So guess what type of information they encode? Resistance to what? Antibiotics. That's the ones we don't like, right? Resistance. We don't like them sharing that type of information, right? So they can, right, unrelated bacteria even, just as long as they can make this connection with one another, and there are plasmids that allow this to happen, that have the information on them to make the sex papillus, right? And sometimes this plasmid, we'll see later, can incorporate it into the chromosome. And so when it does that, and it incorporates, sometimes it can pick up chromosome information and pop back off and be a plasmid by itself. And so then sometimes chromosome information can be moved through this process as well. Right? But we're going to keep it pretty basic, right, in this class, right? Basic steps. But the important thing here is that genetic information is moving, right? And this is laterally, right? So from one cousin to another, you could say, right? Like E. coli to, or Shigella probably gave one of the pathogenic strains of E. coli its toxin, right? So, you know, cousins, right? E. coli and Shigella are kind of cousins. Make sense? Where vertical transmission is when, you know, that cell changes and all its progeny then have changed, right? So we're going down the family tree. In this case, we're hopping trees, right? Make sense? Okay, so let's see, look, clicker. I'm going to walk over to the protein. Okay, so prokaryotes are haploid, therefore mutations usually lead to phenotypic changes. Explain why. Well, there's a couple terms you probably haven't heard in a while, though, so let's start with those. What's haploid mean? One set of, we already said they have one chromosome, right? But within that chromosome, each of the segments of information, what we call genes, right, that code for the proteins that run the cells, they have one copy. We, on the other hand, are what? We're diploid because we have how many copies of all our genes? At least two. One you got from mom, the other set you got from dad. And that's good news for us, right? Because you basically have for the most part, you have a backup copy, right? So especially in the case of sickle cell, some people don't have full-blown sickle cell anemia because they got one good chromosome from one parent and it makes the correct hemoglobin. But you make so much hemoglobin that you actually use both chromosomes. So if you got the other defective chromosome from the other parent, some of your cells become sickle cell depending on which chromosome it picked or how many hemoglobin it made from that chromosome. So you're what we call heterozygous, right? You have one good chromosome, one bad one. And so you have partial sickle cell, and you usually don't realize you have that disease. This disease has been perpetuated, though, because there's an environmental pressure in this world, right? Caused by a lovely little mosquito and a parasite called plasmodium. It causes malaria. Believe it or not, that plasmodium has to infect a red blood cell to replicate. It can't do that in sickle cell. So people who have like partial sickle cell, they're less likely to get malaria, right? The problem is that when you have two people that have partial sickle cell anemia and they mate, you have a 1 in 4 chance with a kid that'll end up with sickle cell anemia, where both parents give both their defective copies to that child. And that's way worse than malaria, unfortunately. But that's why, did anybody know what country or what ethnicity you see more sickle cell in because of more exposure to malaria? Africa and African Americans. Okay, so, but since they only have one copy, unlike us, no backup copy, right? So if it mutates, that leads to disease or defect or difference in them, right? What does phenotypic mean? Physical features. It is the physical expression of the genetic information, right? So in sickle cell, right, that protein, hemoglobin, is the physical expression of your genetic information. Your genome is your code, right, your genetic code. So if that genetic code changes, especially for a bacteria, it's got no backup copy, of course, its protein's going to change, right? It's physical expression of that genetic information. So they change very rapidly compared to us. Make sense? All right. Look, don't have to walk there. Go the other way. I can do this. All right, for the next one, so in order to, bacteria are great for genetic study, for that reason alone, is that we can change them, right? And very rapidly at that. And so in the beginning, they started studying bacteria, right? Because they were so easy to mutate. One chromosome, one copy. This is great. They created what's called nutritional mutants, or auxotrophs. So that means that if you do not give this organism that food or that nutrient, it cannot grow. Right? So these tubes are kind of small, right, on this PowerPoint. And I'll admit that the PowerPoint I'm showing you today is slightly different from the one that's published on Canvas. Because before I zoomed in on this picture, but I want to zoom in on the two that have the boxes around them on this next slide so that we can see it nice and big and I can read it and you can read it. The one thing that isn't right next to me is my water. Which reminds me, if you're in any of my lab classes, right, this is a packet to help you do the homework after midterm. If you want to grab it now today, even if you're taking the test today or tomorrow, you're more than welcome to do so. I didn't have it printed for my very first class, so you didn't have to get it now, but go ahead. While we're thinking about it, while I put my water next to it. The more y'all take, the less I have to carry back to the lab. Okay. So in a lab, right, we could do things like this. So the parental strain, and typically what they would do is, if we're talking about a particular gene, it usually has some abbreviation. In this case, we're talking about the ability to make different amino acids. So they're abbreviated MET. That's a 3-letter abbreviation for methionine, right? And PRO is for proline, right? So in the parental strain, notice it says ++. That means that gene is fine, right? So even though this media contains no methionine or proline, these guys will still grow, right? Notice this color to it. It means that growth happened in that tube. Right? So the bacteria are growing fine because they have the genetic information that even if you don't give them these amino acids, you give them all the other building blocks, they're going to make these amino acids and be able to form proteins. And methionine is very important, right? Because, does anybody remember what's special about that one? There's only one codon for methionine. It's the start codon. So in bacteria, it's the very first amino acid in all their proteins. So yeah, you can't make this. What's going to happen? Well, notice in the next two tubes, right? This guy, right, can't make methionine, but can make proline. But again, can't make methionine, no methionine, cells die. The next one, right? He can make methionine, but can't make proline. It's got a minus there, right? So again, no growth because that media doesn't have proline in it. Both of them die. Where over here, if we have media that contains proline but not methionine, who's going to be able to grow? Right? So it has proline. So if you have a proline defect, you're still going to grow. I got to figure out how to turn that off. Okay? Does that make sense? Well, you see growth there, and you don't in the first mutant. The first mutant can't make methionine, and there's no methionine in that media. It doesn't matter that proline's there. It knows how to make it anyways. It's got a plus. Problem with these mutations, right, is that sometimes they can spontaneously revert back to the normal form. So when they study bacteria, they usually create at least three mutations, so that they definitely can't grow, right? And they can study the effect of that mutation, right? So reversion's a problem, right? So spontaneous mutation, right, correcting back to the wild type, was problematic when they were first studying. So to avoid this problem, right, they do double and triple mutations. So where they'll mutate them so that they can't do two things or three things. But the problem with this, right, there's no problem. What was I going to say? This helps us avoid the problem. I'm all right. I'll get there. Does that make sense, though? Okay. All right. All right. So DNA. So if we're talking about a bacteria, right? Because this is my microbiology class, so we're going to focus on bacteria. We use us eukaryotic cells as examples sometimes, right? Because that makes sense for us. For them, they should have a single chromosome. Sometimes they'll have a plasmid if it's present. If they have all that genetic information, it's referred to as a replicon. You'll see that term. Plasmids, unlike chromosomes, other than that one that we talked about before where it keeps lots of extra copies, that's not normal. Normally when the chromosome is copied, that cell divides, right? So for a normal bacterial cell, one chromosome at a time, a single chromosome. Plasmids, different beast, right? They are smaller than the chromosome, and they can keep multiple copies of the plasmid within the cell and not divide. And so a lot of times when we actually even talk about bacterial strains, we'll talk about copy number. We'll talk about how many plasmids they'll keep. Some of them will keep a very large number of plasmids. Why is this important to us as researchers? Because guess what? We use plasmids as photocopy machines for DNA, right? Although we can do PCR, anyone know what PCR is? Polymerase chain reaction. We're literally in a test tube, right? You use enzymes and heat, cycles of heat and cooling to separate DNA, right? And enzymes to copy the DNA, and then you melt it and separate it again, and you do this process over and over again. You can't do that for really long pieces of DNA, right? There's a limit. Because the longer the DNA gets, the chances are it's going to fold back on itself. In this case, it's a nice circular double-stranded DNA, right? So we can add stuff to this circle, give it to a bacteria, and it'll copy it. It'll make lots of copies. And then we burst open the bacterial cell, and we purify out the plasmids, and then we have huge copies of huge pieces of DNA. It's pretty awesome. What also can be present, which can be a problem sometimes, is bacterial phage. What are bacterial phage DNA? What are bacterial phage, first of all? Viruses that infect bacteria, right? So sometimes they're abbreviated just phage, right? So potentially when you're dealing with a bacteria, at the very least it should have one chromosome, its own chromosome. It could potentially have plasmid. It could have multiple copies of this plasmid. And then it also could have been infected by viruses. And viruses are what we're going to talk about next, right? That infect bacteria.
So does phage DNA affect the plasmid and the chromosome DNA as well?
It kind of depends on what the viruses are saying. So what he just asked was, what if there is viral DNA in there? Can it affect the chromosome or the plasmid? And it can affect either. It just depends on what type of virus you're dealing with. And so we'll get to that. So there are three ways in which plasmids differ from bacterial chromosomes are important as we continue our genetic discussion. So anyone remember one thing, just in the picture alone, size. Plasmids are smaller, right? Plasmids are smaller. Do all bacteria have plasmids? No. Do they contain information that a cell has to have? Of course not, because not all of them have it, right? So they're smaller than chromosomes, right? They code for non-essential information, which means you don't have to have it. But they're kind of like bonus, right? Because they could code for stuff, right, that we don't necessarily want them to have. So they frequently occur in far more copies, too, than the chromosome. And they can be transferred from unrelated bacteria, unlike chromosomes. Right? Chromosome information, really you have to be closely related for that information to be shared. So you need to find a spot to put it, right? If you're not closely related, there really isn't a place to put that information, right? In that file like cabinet of DNA. All right. So now we're gonna. Okay, so DNA changes. We get mutants. How do we find these mutants? How do we isolate them? How do we study them? One way is direct detection, and an example of that is antibiotic resistance. We use this a lot in the lab. Tends to usually freak out my students when I tell them this, And we're going to do this in microlab, y'all, at the end of the semester. Lab 12. We're going to make fluorescent green E. coli. How are we going to do that? We're going to literally put DNA from jellyfish into the E. coli. And they'll express that protein, and under UV light, they're going to fluoresce green. But to select for them, the other thing we give them is antibiotic resistance. So only the ones that take in the DNA and are resistant to the antibiotic will grow on the plates that we use, which contain antibiotics. Right, so that's one way, and that's one way researchers are trying to find bacteria that produce antibiotics. Because microorganisms, bacteria, make these against each other, right? So one of the ways that we find out if they make it is we literally will take one bacteria and grow it in the presence of the other one and see if they are affecting each other. It's pretty amazing stuff. And then, of course, we hope that one's producing an antibiotic, and we isolate it and study it. We hopefully get a new antibiotic. So antibiotic resistance, that's something we can see, right? That we can physically measure. We can directly detect that organism has changed. But sometimes, especially with the first mutations that we talked about, those auxotrophs, if we didn't give them the nutrient they needed, what happened to them? They died. Well, how are you going to study something that's dead? It's like, oh, I just found out this one can't do proline, but I killed it in the process. Crap. I'm not going to study it, right? Then you got to do things like indirect detection, or what we call replica plating, right? So, I keep going the wrong way on this thing. It doesn't make sense to me. Okay, so phenotypically, they phenotypically change, right? Especially if they're like changing color, say, right? Their morphology, their growth on the plate, that's great. We can directly detect them. In this case, right, they ended up, whatever mutation it was, they were resistant to streptomycin. So only those cells growing on that plate are the ones that are resistant to streptomycin. So we can directly, right, those are the only ones we're dealing with, because the other ones were what? Killed by the antibiotic. So those are all just your mutants there, growing on that plate. And their mutation happens to be, one of their mutations happens to be, they're resistant to that antibiotic streptomycin they use. So phenotypic changes, right, where we can physically see, those are great. But what if, right, it's something that we can't detect or we detect by killing them? So in the second screening, you all see how the bacteria are put on particular places on that plate? Right? That's one similar to a method that I'm going to show you from your book, because I haven't updated my PowerPoints from the new edition of the book. But in the past, this used to be a very common method of doing indirect detection of mutants, is replica plating. And so this is some container with sterile felt on it, right? You guys know what I mean by felt, that fuzzy material? So it's sterile, so there's no bacteria on it. And basically what they're using it as is a stamp. What I don't like about this picture is typically the scientists would draw a line on the plate so that and on their stamp so they match it up. So when they stamp it, it's always stamped in the same orientation. So you have your master plate, right? And everybody is growing. And then you have different plates that you've stamped it on that have different growth conditions. So say, for instance, this one doesn't have proline. And so then you'll notice you're missing these two colonies, right? And so you know, oh, they can't produce proline. But we killed them, right? They're gone, right? That's empty circles on that plate. But you still have your master plate, and you know the location of where they were, so you can just pick them off the master plate and then continue to study them. So now I have to walk over to the podium because I want to show you a technique that's more oftenly used now in labs than that one. So in your book, which I already opened, is this type of replica plating. So they'll have bacteria, and they'll either have petri dishes with grids on them, or they'll put them on top of a grid, and they literally take toothpicks, because they're going to inoculate a lot of squares. I've never done this many squares, but, some scientists will do that many squares. Well, they'll take the toothpick, and they'll inoculate a whole series of plates, right? One master plate where everybody will grow, and then other plates with, different types of media on it, and you can see the differences, right? So you'll see, right, different spots where there was no growth, and you can just go back to your reference plate and get those organisms to study them. So that's a more widely used form of replica plating. There's this fancy tool that somebody came up with once. It scared the crap out of me, though. It looked like a whole bunch of needles, right? Basically, had a whole bunch of needles sticking down, so you could stab your plate and then stab it onto other ones and inoculate that way. Turned out to not be as effective as these lovely sterile toothpicks. That's drop my clicker. We'll find the lid later. Hopefully I don't electrocute myself. I'm trying to get, it's right over my, there we go. Okay, so, let's see if it even works. Yay! Dropping it isn't breaking. Okay, so this one is not covered in your book. But this is an important technique. This is the Ames test, of course, named after someone named Ames, right, who came up with it. And so this helps potentially detect mutants. And so why would we, so let's think about this in our everyday lives. Why would we want to know if a chemical mutates? Well, one, it could be a possible carcinogen if it mutates DNA, right? So you're in the cosmetic industry, right? And you just came up with this new face cream, right? You think it's going to get rid of all the wrinkles that ever existed. Looks great. You going to put it on your face right away? Yeah, I'm certainly not doing that. So this test has been done for years on chemicals and things like that, where we expose the bacteria to these chemicals. And of course we have a control group, and it uses actually a specially mutated strain of, I'm not going to say what bacteria because I'm going to get it wrong, and I want to be wrong. But it can't produce histidine, so normally it won't grow. When there's no histidine, it won't grow. But if it mutates, it'll grow, right? So that's not good. Right? And so this has been done for years, right? We've been exposing bacteria to chemicals, right? See what happens, see if it mutates them. Okay. So the good news is if your cosmetic passes the Ames test, then it goes on to step 2, which most people don't like to talk about. Animal testing, right? The cute and fuzzies, right? I don't know about you, but again, I'm not sticking it on my face until I know that a cute and fuzzy didn't, die from it. But what's the good news is that nowadays we're developing the ability to test things in tissue culture, right? So we're almost to the point where we can almost fabricate the body in a petri dish, right? And test these chemicals in a much safer way instead of exposing other animals to it. So our technological advances have been going, but this has been going on for a long time. No one cares that we, torture poor bacteria. They're not as cute and fuzzy, right? That's the problem, poor bacteria. So we've covered most of this objective from the last chapter. But transposons and knockout mutations were not covered in that chapter. They're covered in this chapter. And so transposons are sometimes called jumping genes because they literally move. Was discovered by, in this case, a woman. So give props to her, Barbara McClintock. This is her and her lovely microscope, right? So these are movable elements, right? So sometimes called jumping genes. So they move throughout the chromosome. That sounds all well and fine, right? So they move. What's the big deal, right? And what got her to think about this and to investigate this? And you want to talk about a meticulous scientist, which is pretty impressive. She looked at maize corn, and most of the time y'all didn't have a clue what I was talking about when I said that. So that's a picture of it, y'all. Sometimes referred to as Indian corn. You see how the kernels are different colors? Those different colors are due to jumping genes, right? And what happens is when the genetic information moves, it disrupts other information. In this case, either producing a different color or preventing a color from being produced. So what we have here in the big circle, right, could be a plasmid, could be the chromosome, doesn't matter, it's DNA. And DNA is divided into different segments. Those are genes that code for information. So say we have gene 1, 2, and 3. And they are a transposon. They're a jumping gene. That section can move, right? So it moves to, say, the nearby plasmid. But when it jumps into that plasmid, notice it inserts into B. B is a gene that codes for something, right? But RNA polymerase can no longer make B because it's been split up by that jumping gene 1, 2, and 3. So it's essentially knocked it out, right? Made B non-functional. It'll make 1, 2, and 3, right? They're intact. But we just disrupted B. B is non-functional now. And they've literally done this to the organism level. When I worked in research, I didn't understand why a particular group of mice in the animal house was, got such special treatment. They were knockout mice. And one of the premier immunologists that studied there at LSU Vet School, those were her mice. And they have been genetically engineered to have a particular gene ability knocked out. Right? So that mouse couldn't do something. Right? Didn't have that gene anymore. So that she could do a particular immunological study on those mice. So yeah, as you can imagine, at that level, that's very expensive. Bacteria, not so much, right? We do this kind of stuff all the time. Right? I did this in my research even. They're probably doing it in high school labs nowadays. It's become so cheap. So this is a real example of doing that. And scientists like myself use this system to, again, just get bacteria to copy DNA, and then I sequenced a couple of genes from a couple of different frogs. I'm going to skip over this specific example for right now and hope that we can get back to it later. But, we've lost some time thanks to crappy things like weather and other conditions. So maybe we'll come back to it. But you can read about it in your book, and if you have questions, definitely ask me. So we're going to move on now to all these forms of horizontal, or what's sometimes referred to as lateral gene transfer. So this is the ability, to go from cousin to cousin, E. coli to something like Shigella, or vice versa. And so we've already talked about this one. This was the very first objective in our genetics topic we started. Anyone remember whose experiment with the mice and trying to develop a vaccine for Streptococcus pneumoniae actually led to discovering transformation? Griffith experiment, right? It was the Griffith experiment. Okay, so we'll talk about it again, right, because that's transformation, and transformation is lateral gene transfer. We looked at and opened up our lecture today with conjugation, so we'll just kind of review that. And then we'll add in transduction, and then, oh my goodness, I just said 2 words that start with trans, right? And so trans really means to move, right? Right, transit, movement. Transformation, right, really allowed the organisms to change, right? They were able to produce that capsule that they didn't produce before, right? And microbiology lab, we're going to transform them into glowing green E. coli, right? So it's a process, right, where you can literally change the phenotype of how that organism looks. Right? So I always tell my students to think about that, or think about transformers, right? Right? Transformers go from those cars to those alien robots. My son is finally old enough that he does understand that that's fiction, and whoever's driving that Camaro around town is not Bumblebee. That would scare the crap out of Mom. That turned into an alien robot. But can you imagine what we mean when we say transformation? Because now when I say the word transduction, I want you to focus on the duction part, right? On a duct. What is a duct? It's a duct. Stuff moves through. It's like a pipe, right? Duct is something things move through. So in this case, the DNA is going to move through something. Anyone know what it's going to move through? Not conjugation. There's something that's going to literally pick up the DNA from one virus and bring it to another one. I said it. It's a virus. It's going to pick it up from one bacteria and bring it to another bacteria. But it is a virus. It's a bacterial virus that's going to do that. Yeah, bacteriophage. Yep. Accidentally giving you this. So this is just another drawing of the Griffith experiment, right? Where the cells, right, the living cells have the capsule that kill the mouse. The non-encapsulated living cells, because they don't have a capsule, they don't kill the mouse. But when you put the killed, right, so just because the cells are killed, the difference here is they still have their DNA, right? So you kill those bacteria, and you basically just put their DNA in the mouse with the living non-encapsulated cells. The living non-encapsulated cells picked up the DNA from the encapsulated cells and learned how to make a capsule and killed the poor mouse, right? So when he autopsies and he gets back out, right, encapsulated bacteria, how did they learn how to make that capsule? Remember, anything that a cell knows how to do is encoded in its DNA. So these got DNA. At the time, remember, this is way back before we even knew it was DNA. But he knew something was transforming. So for transformation, right, it doesn't require cell-to-cell contact, right? They just have to be in the general vicinity of one another. The bacteria can be naturally what we call competent, and this is that ability to pick up DNA from the environment. Not all bacteria have this ability. It's actually a quite complex process. A lot of things that you have to be able to do this. But we can purposefully make them competent. We can use calcium chloride ions to help shield DNA and shove it into cells. We can do it with electroporation even, right, where we use electricity to shove DNA into cells, whether it be bacteria cells or eukaryotic cells. So although this process happens in nature, we can cause it to happen in the lab. Right? We can do it unnaturally. So, in order for it to happen, right, DNA has to be in the environment. it has to attach to the surface of the cell. So these cells that have this ability, these competent cells, right, that are naturally competent, they have these protein complexes in their cell wall that allows them to attach to DNA in the environment. If the DNA is double-stranded, it actually strips away one strand and the other strand enters into the cell. And then it has to find a place to put it. And this is why this typically only happens, the natural form of transformation only happens with organisms that are closely related to each other. Because you got to find some place in the chromosome. They literally have to trade out information. And they're going to say, okay, I'm going to trade what I just picked up for what I have right here. It's similar, so I'm going to trade it out. And of course, you know, these aren't thinking beings. They're not thinking, right? But these molecules, right, it has to have this matching up. And if it does happen, then they could potentially be transformed, have a new ability they didn't have before. I don't have a picture I want. We'll come back to it. Okay. Conjugation. Conjugation can be quite complex if you read in your book, right? You're like, oh my Jesus, Erica, give me the short version. I'm going to do that. Okay. So, as I mentioned in our opener, right, there are special plasmids that allow conjugation to happen. They're called F plasmids for fertility. On that circular piece of DNA is the ability to make that sex pilus. So only bacteria who have this plasmid can do conjugation. But then once they pass it to somebody, right, once they give some other cell this plasmid, they too can do it. So when they don't have it, they're termed negative, right? And when they get the plasmid, they're plus. And sometimes they will lose these plasmids. It takes energy to maintain this information, to copy this information. So sometimes if there isn't selective pressure to keep it, right, if it isn't beneficial, they will get rid of it. And so they can actually go from plus having something to minus not having it. But typically, right, they're going to have it and they're going to pass it on to others. And this does require direct cell-to-cell contact, right? So they're going to hook up with each other, right, and through that sex pilus, the plasmid, one strand of it goes through, and it gets the complementary strand gets copied on the other side in the previous slide, and it makes it double-stranded again. That's the simplest explanation of it, right? As I mentioned in our opener, sometimes those plasmids can interact with the chromosome and pick up information from the chromosome, right? That gets really complicated. What I want y'all to understand and remember about conjugation is that you definitely have to have that physical connection, right, through the sex pilus, and that is from having at least this special plasmid, the fertility plasmid, that has that information on how to make the sex pilus on it. That's the most common thing they're going to share, and on that plasmid there could be other things, and they can share other plasmids when they can do this process, right? So this information, they're hooked up to each other, right? They're directly sending it through a very safe protein tube. The DNA is protected. That's important because, you know, things like transformation, the DNA is in the environment, right? And it could be damaged. So transduction, on the other hand, right, sometimes during phase replication, when that virus gets into the bacterial cell, instead of packaging its own DNA into its protein coat, it packages the bacteria's DNA. Because what happens is when it infects a bacterial cell, these bacterial viruses, is they cut up the bacteria's chromosome. Because they don't want that bacteria cell to be a bacteria cell anymore. They want that bacteria cell to be a virus factory. And so they're going to use all the resources and machinery inside of that cell to make more viruses, right? But accidents happen, right? And that's how this happens. But again, that information, when it's brought to the next virus, it has to have a place to go. That information has to have a place to go. So here is that lovely little spaceship guy, right? That's a bacterial phage. That's a virus that specifically infects bacteria. It's all that you see, that spaceship-like thing is the protein coat, and then you see the DNA is injected into the cell. So just the DNA goes in, and any enzymes they may need that they bring with them. And they're going to chop up, right, the bacteria's DNA. So now they're going to make their viral protein coats, they're going to make their viral DNA, and they're going to package these things up. But you can see in a couple of them, right, they've got the blue DNA, that was to denote bacterial. So then they go and they infect, and viruses have what we call host ranges, right? Viruses that only infect certain bacteria, just like their viruses only infect certain animals, right? And this has to do with what we're going to talk about when we talk about viruses next, is the very first thing that has to happen is that virus has to attach to that cell. So there has to be something on that cell that that virus recognizes. Okay? So this is going to happen among cells that are similar, right? That can be infected by the same virus. So the virus is going to inject that DNA. It's not viral DNA, it's bacterial. It can potentially incorporate it into that chromosome, and then that bacterial cell has been transduced. Right? It's gone through the process of transduction, and therefore has genetically changed and probably phenotypically changed. So, why do that, though? Why pick up DNA? Like, from the environment, why do those bacteria pick up DNA? Why would you join with another bacteria and exchange DNA? DNA is information, right? Information allows you to do things. And remember, their chromosome, one copy. What happens if it gets damaged? It's important stuff. They could what? They could die. So some scientists believe this is like kind of their way of sexual reproduction, or it probably started out as a way of potentially being able to repair their own DNA. When their DNA gets damaged, right, if they can do transformation, they could pick who's dead. Probably their friends around them, right, that hopefully their DNA didn't get messed up, and they can pick it up and patch their own. Right? Same conjugation, really, that one is more like, okay, this is more on the lines of sex, of, exchanging DNA. But it's not necessary for them, right? They don't have to conjugate, right? But they obviously can benefit from that, right? If you can get information from another bacteria, that could be beneficial for you. And the scary thing for us with conjugation is the cells don't have to be related, right? With transduction, right, infection with a virus and transformation, that DNA has to come from relatively related bacteria because it definitely has to incorporate into the chromosome. With conjugation, it can be just a plasmid, right? Sometimes that plasmid might contain chromosomal information. It doesn't have to. What time is it? I have time. I know y'all hate that, right? Don't tell me that. Okay. Back to the switch. Okay. So, this, of course, is plasmid. Right? This circular thing represents a plasmid. And all DNA has to have a site where replication is going to start. That's the ORIV site, right? That's the origin of replication site. So that's where the enzymes attach and you can copy the information. So you'll notice that AMP, AMP, R. Amp is ampicillin. That's an antibiotic, right? So on this design plasmid is an enzyme that makes the bacteria resistant to ampicillin. So we can do direct selection. So if the bacteria do not take in this plasmid and we grow them in the presence of ampicillin, they'll die. If they have this plasmid, they'll grow. So that's one way we can select for what we call transformants. This is going to be a process of transformation, but shoving a whole plasmid in there. Right? Okay. On the plasmid is another gene called the LAC-Z gene. This makes an enzyme that can digest lactose. It's the LAC. Within that gene, there are multiple what we call cloning sites that restriction enzymes can cut. So you notice this right here? See all those abbreviations? Those are for restriction enzymes. Guess who we discovered them from? Bacteria. It's like their own little immune system. These are special enzymes that only cut DNA at specific codes. Right? So the enzyme sees this code and it cuts it. So if we take DNA that we have and we cut it with that enzyme, and then we take this plasmid and we cut it with the same enzyme, and we put that cut plasmid and the pieces of DNA we want together in a test tube, they'll join, right? Because they match, right? It's like cutting with those zigzag scissors, right? Those craft scissors. We cut both pieces of, we can eventually put the pieces back together, right? Because they were cut the same. That makes sense? Okay, so that's how we put extra, but when we do that, right, we're disrupting this gene, right? If we put stuff right here, we're disrupting this gene. Can it make that enzyme anymore? No, right? And so when we grow it with this chemical, right, Xgal, right, if we don't disrupt the gene, right, so that's what's shown here, we don't disrupt it, they produce the functional enzyme, right, beta-galactosidase. It can break Xgal, right, and they'll grow blue. You'll get blue cells. That's bad news. That means they took in the plasmid, but the plasmid didn't pick up the DNA we put in the tube, right? Instead, what we want is the white bacteria, because one, it tells us, because they're growing in the presence of ampicillin, it has the plasmid, and two, it has whatever DNA we inserted into that plasmid. So then we pick up those white bacteria, right? We grow them, we amplify them. They make copies of that plasmid for us. We lyse the bacterial cells, we take that plasmid out, and we can do things like sequence it, and cool stuff like that. So I literally did that as a graduate student. It's not that hard, y'all. It's just like all of the instructions. So I'm not kidding you when I think that probably at this point high school students are doing this kind of stuff. It's not that hard, right? But you need to understand the premise of what's going on here, right? So they can't do something because we disrupted that DNA. Because we stuck DNA and we wanted them to copy for us. They're great copy machines. They're like way better than the copy machine. Because remember, what's the rate of mistake? 1 in a billion. 1 in a billion that they're going to make a mistake. Okay. All right. So we will end there.